So good morning, everybody. My name is Georg Schulz. I'm from the University of Basel, from the Department of Biomedical Engineering, and there from the Biomaterials Center. And today I'm going to talk about non-destructive three-dimensional analysis of peripheral nerves. So at the beginning of my presentation, I would like to present you some of the topics running at our center, at the Biomedical Science Center. So one of the topics is nice science for human health. The previous talk Andreas gave just right now already showed these shear-sensitive liposomes. So what I just wanted to add here is that we are doing the imaging of the arteries in order to simulate or to make flow simulations and to get an idea about the shear stress available at the stenosis. So here's for example, a micro CT phase contrast image of the artery where the lumen was segmented, and then using this segmentation of the lumen, you can perform flow simulations and then get an idea about the shear stress in the stenosis. Another topic is, for example, dental research. Here it contains investigations of teeth nanostructures. Oh, this is far. So where we perform small angle X-ray scattering and analyze the collagen in tooth. Then we analyze the precision of oral scanners, tooth repair, smart implants, so shape memory alloy implants, bone graft materials, and bone scaffolds. Another topic is cancer and neurodegenerative diseases, where we, for example, do tumor imaging, again, high resolution, so microscopic tumor imaging, where we again extract the blood vessels and then do vessel analysis and compare the vessels within the tumor to the vessel tree in the healthy tissue. Then brain imaging, where we analyze sclerosis or stroke, cell visualization. This is a micro CT image. This is the histology slice of it. And also nanostructures of brain, again, small angle X-ray scattering, where we investigate myelin within the brain tissues. And this is, in principle, my field, so the X-ray imaging, the high-resolution X-ray imaging. And here you see some of the projects where I'm working on. So again, the oral scanners, we have projects with the Natural History Museum, knee anatomy, tissue engineering. We go to synchrotron radiation to use the X-rays for the micro CT experiments, and of course have the lab CT available. And this is one of the core facilities at the Department of the Biomedical Engineering, where we offer scans to other research groups and companies. So now I would like to start with the talk. And at the beginning, just a really short introduction to the principle of the computer tomography. Here you can see a typical setup of computer tomography, where you have the sample, which is mounted on the high precision rotation axis. From the left, you have the beam, a detection unit. One computer acquires the data, and then you have a second computer where you can reconstruct. So you acquire images at different rotation angle positions, around 1,000, 2,000 images, and create such a sinogram where one line is one image, like one angle of the whole scan. And using this, you can reconstruct the data and have virtual 3D information of the specimen. So this is one of the devices we have in our system, in our lab. And also the data I will show were acquired by this system. This is the nanodome from GE. Here are some parameters. I don't think I have to explain everything, but one of the main things is the spot size. The spot size is in principle the limitation of the spatial resolution we get. So it's around 800 nanometers resolution you can obtain, but isotropically and in 3D. And what else is more? The rest is probably not so important for this talk. Now we come to the sample preparation. So during the sample preparation, first nerve conduits were produced using spinning mandrel technology, which you can find in this publication. It's profound. Then left sciatic nerves in rats were surgically operated, which created 10 millimeters of gap. And this gap was bridged then by the nerve conduits. And later, after 12 weeks in this case, were then extracted again. And for the comparison, normal nerves were collected from the other side of the same animal. After the extraction, the specimens were paraffin embedded for further experiments. And the experiments were done again with the system I've just shown, with a 60 kV acceleration voltage and 280 microamperes current with a pixel size of 4 micrometer. These two images show you the healthy nerve on the left side and the diseased nerve on the right, which is around 10 millimeters length in principle. This is a little longer. And I'm not sure if you see the arrow. So here's the one red color. The red color arrow is the surface growth between the major nerve fascicles. The yellow arrows here indicate the two ends of the reconstructed nerve. So the nerve conduit was placed here. So here's the healthy nerve. Here's the repaired nerve in principle in this region. And then you have two zoom-ins of these small boxes here to get a feeling of the structures visible. So in principle, what one can see is that more muscle fibers are seen on the nerve surface, so the white colors here. So more muscle fibers are seen in the healthy nerve compared to the regenerated nerve, and one can calculate the mean diameters of the nerves, or the muscle nerves, which are around 50 micrometers. And if you look in the zoom in, then you can see that the healthy nerve has a much higher parallel arrangement of these structures here. So one of the main advantages of computer tomography is that you can cut through the specimen in any direction you like. So it's not like histology where you just cut once and that's it. You can virtually cut in all possible directions. Here you can see again the healthy nerve and the diseased nerve on the bottom, cross sections, and then orthogonal cuts through it. So there should be a line where this cut is located in the cross section. And the scale bar here corresponds to 1 millimeters. If you have a look on the histogram of the 3D data sets, so histogram means these black dots in this graph correspond to specific, or the peaks in the graph correspond to specific attenuation value or to specific structure of the specimen. So the first peak is air, the second peak is the paraffin, and then this peak corresponds to the nerve tissue, And what you see here is on the left-hand side, again, the healthy and the reconstructed nerve, that the attenuation values of the reconstructed nerve are shifted to the left. So you immediately see that by looking on the attenuation values, which one is the reconstructed nerve. Further on, one could calculate a cross-sectional area. So in principle, the area of the nerve at each position in this case. And the blue one is again the healthy one. And there you see that the cross-sectional area is very constant over the long distance. Then you have here the reconstructed nerve, where you see that at the beginning and at the end you have the higher values, higher area values, which is in principle the position where the conduit was fixed. So afterwards is again the healthy nerve, and in between was the reconstructed nerve. And then there is a comparison of some geometrical parameters on the right-hand side, where we had a look on some values like median cross-section, average cross-section, maximal cross-section, again between the healthy and the reconstructed nerve, where always the reconstructed nerve had higher values. Another feature of X-ray tomography is that one can segment or make tissues semi-transparent or transparent. In this case, we made the tissue transparent and had a look on the blood vessels of the two nerves. So so-called intensity threshold. And to give you a feeling, so these two vessels here, you can see have diameters of 0.04, so 40 and 20 micrometers. And if you have a look in the reconstructed nerve, then you see that the arrangement of the blood vessels is more heterogeneous. So you have much more homogeneous arrangement of the blood vessels in the healthy one. And also the density, the vessel density is higher in the healthy nerve. So you have much higher number of vessels inside the healthy nerve. So with this slide, I would like to go to the conclusions. So I've shown you a method to 3D visualize regenerated nerves or healthy nerves with true micrometer resolution in all the 3 dimensions, so isotropic. And after this, you still have the chance, so it's non-destructive, therefore you can just go on and use this same specimen for further analysis like histology. So I've shown you the micromorphology of the nerve tissues, and the results were validated with a sufficient number of samples to provide findings with statistical significance. And on one of the last slides, I showed you quantitative parameters where we compared the healthy and the reconstructed nerves. This slide, I would like to thank the Swiss National Science Foundation for the investment, for the and the people on the manuscript who were working on this project. And with the last slide, I would like to thank you for your attention.
Thank you very much, Georg, for going so deep into the nano world. Talk is open for questions. Can you please step out to the mic? Sorry.
A very interesting talk. Thank you very much. I was interested in the minimal resolution of the nanotome and the required time it takes then. Once you had 4 micrometer, I guess that also due to time reasons. So how long can you go down? And you're using X-ray. Does it damage also healthy material? Are they still live then or not?
Yeah. So first to the second part maybe, because it's so It doesn't change anything. So if it's especially this tissue is embedded in paraffin, there's no problem with the X-rays. Also, most of the X-rays pass by. Only some of them are attenuated by the specimen. And about the scan times are between 15 minutes for like normal scan, fast scans, where it's pretty noisy, but still you can see something. And if you go down the resolution, then you could get scanning times of around 10 hours. So the smallest pixel size you can get is 200 nanometers. But as I told, the pixel size is not yet the resolution, so the resolution is limited always by the source size. So there's 4 different modes of the scanner where you adjust the source size. And of course, the smaller the source, the less photons you have, the less X-rays you have. And the longer will be the exposure time. So up to 10 hours you can take. But if you would calculate the time you need to cut the whole nerve and stain every slice to get the same results, and also you will never get the third dimension because it's maybe 10 microns, or depending on the cut thickness of the histology. So I think it's feasible 10 hours scan time without interaction. So you put it in and then it's running.
Thank you.
Yeah.
Are there other questions? If not, then thank you very much, Georg.
Thank you.